Hello everyone, once again welcome to my channel. This is Dr. K. Srinivas Reddy, Assistant Professor of Botany, Government Degree College for Women, NALGONDA. In my last video, I have explained about nuclear pore complex. In this video, I am going to explain nucleic acids. Okay, the topic nucleic acid is the part of your fifth paper, that is cell biology and genetics of your fifth semester, and it is very important for your academic examinations and also competitive examinations which you are going to appear after your graduation. Okay, let us come into the topic nucleic acids. Nucleic acids are complex molecules and they possess genetic information, and nucleic acids are large molecules. Nucleic acids are complex, informational, and macromolecules. Nucleic acids possess genetic information. Okay. Nucleic acids are found in all living cells and viruses. Nucleic acids were first isolated by Joseph Frederick Mischer in the year 1868. Joseph Frederick Mischer isolated the nucleic acids from the nucleus of pus cells from the wounded soldiers, and he observed high content of phosphorus and nitrogen in these substances. The substances which isolated from the nucleus contain high amount of phosphorus and nitrogen, and he named these substances, he named these phosphorus rich substances as nucleins, since they are separated from the nucleus. As they are separated from the nucleus, he named these substances as nucleins. Later on, Altman coined the term nucleic acid for these substances because these substances are acidic in nature. By the 1950s, scientists have observed two types of nucleic acids. They are DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Cosell identified ribosugar and nitrogen bases in nucleic acids. Cosell identified ribosugar and nitrogen bases in the nucleic acids. Both DNA and RNA are large molecules and occur either in single or double-stranded states. DNA and RNA occur either in single-stranded or double-stranded. In some viruses, single-stranded DNA is also present. Usually DNA is double-stranded, but in some viruses, single-stranded DNA is also present. Double-stranded RNA is also present in some viruses. So that is the reason why DNA and RNA occur either in single-stranded or double-stranded states. Okay. Let us discuss about the chemical composition or constituents of the nucleic acids. Let us discuss about the chemical constituents of nucleic acids. Chemical analysis have shown that nucleic acids are composed of following three compounds. Number 1, phosphoric acid. Number 2, pentose sugar. Number 3, nitrogen bases. Nucleic acids chemically composed of three compounds. Number 1, phosphoric acid. Number 2, pentose sugar. And number 3, nitrogen bases. Two types of nitrogen bases are present in the nucleic acids. They are purines, and pyrimidines. Chemically, nucleic acids composed of three main compounds. They are phosphoric acid, pentose sugar, and nitrogen bases. Nitrogen bases are again two types, purines and pyrimidines. Let us discuss all these compounds in detail one by one. Okay. Let us coming to the phosphoric acid, one of the components of nucleic acid. The chemical formula is H3PO4. H3PO4. Phosphoric acid contains 3 hydrogen atoms, 4 oxygen atoms, and 1 phosphorus atom. Phosphorus is a pentavalent ion and it can form 5 bonds with other molecules. Here you can see in the picture, this is the phosphorus atom, and here you can see 5 bonds. Here you can see 2 bonds, 3, 4, 5. Phosphoric acid has 3 OH groups, 1, 2, and 3. Phosphoric acid has 3 OH groups or hydroxyl groups, of which 2 hydroxyl groups are involved in the formation of sugar phosphate backbone. Phosphoric acid has 3 hydroxyl groups, of which 2 hydroxyl groups are involved in the formation of sugar phosphate backbone. Phosphoric acid or phosphate moiety binds to the 5th carbon of one pentose sugar and the 3rd carbon of the other pentose sugar of DNA and produce phosphodiester bond or phosphodiester linkage. Here you can say very clearly, this is the 5th carbon of one pentose sugar, and this one is the 3rd carbon of other pentose sugar, and this is the phosphate moiety. Ester bond is formed in between 5th carbon of one pentose sugar and OH group. Hydrogen is removed from this hydroxyl group, only oxygen is present here, and this is the 2nd ester bond, which is also present in the 3rd carbon and in between other OH group of phosphate moiety. Here also hydrogen is removed. So in this way, 2 ester bonds are formed in between phosphate moiety and 2 pentose sugars. So in this way, sugar phosphate backbone is formed due to phosphodiester linkage. Sugar phosphate backbone is formed. Okay. Let us discuss about the second component of nucleic acids, that is pentose sugar. Coming to the second component of nucleic acids is pentose sugar. There are 2 types of sugars present in the nucleic acids. One is ribose sugar and second one is deoxyribose sugar. 2 types of sugars are present in the nucleic acids. One is ribose sugar and the second one is deoxyribose sugar. Based on the sugar found in the nucleic acids are of 2 types. One is ribonucleic acid and the second one is deoxyribonucleic acid. Ribonucleic acid RNA, deoxyribonucleic acid DNA. In RNA, ribose sugar is present. In DNA, deoxyribose sugar is present. In the picture here you can see both types of sugars. This is ribose sugar and this is deoxyribose sugar. Either deoxyribose sugar or ribose sugar, only one is present in the nucleic acid, both are not present in the same nucleic acid. Okay, and ribose and deoxyribose sugar both are same in almost all in chemical structure. Ribose sugar if you see the ribose sugar, which is a 5-carbon compound, first carbon is present here, second carbon is present here, third carbon position is here, fourth carbon position is here, and this is the fifth carbon. And here you can see 5 oxygen molecules are present in ribose sugar. This is the first oxygen molecule, second oxygen molecule, third oxygen molecule, fourth oxygen molecule, and fifth oxygen molecule. If you see the chemical formula of ribose sugar is C5H10O5. Coming to the deoxyribose sugar, the chemical formula of deoxyribose sugar is C5H10O4. Here O5 in ribose sugar. In case of deoxyribose sugar, only O4. That is the only difference in between these two sugars. If you see the structure of ribose sugar, on second carbon of ribose sugar, OH group is present. Here you can see this is the OH group. In the same place of deoxyribose sugar, you can find only H on the second carbon. In place of OH, only H is present. So that is the only difference in between these two molecules, in between ribose and deoxyribose sugar molecules. Only difference is one oxygen. One oxygen is more in ribose, whereas less in deoxyribose sugar. So that is the reason why deoxy, less number of oxygens, that is on second carbon. Here you can say 2 deoxyribose sugar. 2 deoxyribose sugar. Ribose sugar is present in RNA. Deoxyribose sugar is present in DNA. Okay, now let us discuss about the third component of nucleic acids, that is organic bases or nitrogen bases. Okay. Coming to the third component of nucleic acids, the organic bases or nitrogen bases. The organic bases present in the nucleic acids are heterocyclic compounds containing nitrogen in their ring. Hence, they are called nitrogen bases. Organic bases present in the nucleic acids are heterocyclic compounds containing nitrogen in their ring. Hence, they are called nitrogen bases. Nitrogen bases found in the nucleic acids are of two types. One is purines and the second one is pyrimidines. Two types of nitrogen bases are found in nucleic acids. One is purine and second one is pyrimidine. Let us come to the purines. Purines are derived from their purides, and purines contain 2 CN rings. Purines contain 2 CN rings in their structure. One is 5-membered pyrimidine ring, and another one is 6-membered pyrimidine ring, and second one is 5-membered imidazole ring. Purines contain 2 carbon nitrogen rings in their structure. One is 6-membered pyrimidine ring and second one is imidazole ring. There are 2 types of purines found in nucleic acids. One is adenine or 6-amino purine. Second one is guanine or 2-amino 6-oxypurine. Here, if you see the structure of adenine, adenine consists of 2 CN rings. This is the 6-membered CN ring, which is a pyrimidine ring. It looks like a benzene ring. Nitrogen is present in first and third position in place of carbon. Nitrogen is present in first and third position in place of carbon, and one amino group is present on 6th carbon. Amino group is present on 6th carbon. Coming to the imidazole ring, nitrogen is present on 7th and 9th positions in place of carbon. This is the imidazole ring, okay? Imidazole ring, which is a 5-membered one. In this ring, On 7th position and 9th position, nitrogens are present in place of carbon. Adenine is also known as 6-aminopurine. 6-aminopurine because on 6th carbon of pyrimidine ring, amino group is present. Amino group is present on the 6th carbon of pyrimidine ring. Hence, it is also known as 6-aminopurine. Coming to the guanine, guanine also consists 2 CN rings. One is 6-membered pyrimidine ring and 5-membered imidazole ring. Like adenine, on 1st and 3rd positions, nitrogen is present in place of carbon. On 2nd carbon, one amino group is present. On 6th carbon, one oxygen group is present in the form of keto group. Right? On adenine, amino group is present on 6th carbon, whereas in guanine, oxygen is present on the 6th carbon. In guanine, on 2nd carbon, amino group is present that is missing in adenine. So this is the difference. Amino group is present on the 6th position in adenine, whereas here amino group is present on the 2nd carbon. On 6th carbon, keto group is present. Adenine and guanine are the purines present in nucleic acids. Adenine always pairs with the thymine with 2 hydrogen bonds, and guanine always pairs with cytosine with 3 hydrogen bonds, or vice versa. And this is called the pairing is called complementary base pairing. Coming to the pyrimidines, pyrimidines consist of only one ring, that is 1 CN ring, that is pyrimidine ring. And there are 3 types of pyrimidines present in nucleic acids. One is thymine, second one cytosine, third one uracil. Thymine and cytosine are found in DNA. Cytosine and uracil are found in RNA. Thymine, in place of thymine, uracil is present in RNA molecules. Thymine is not found in RNA. If you see the structure of thymine, it is also known as 2,4-dioxy 5-methylpyrimidine. On 2nd carbon, oxygen is present, and also on 4th carbon. Here number starts from here 1, 2, 3, 4, 5, 6. Whereas here in case of pyrimids, number start from here 1, 2, 3, 4, 5, 6. The numbering of carbon is given based on the functional groups present on the CN ring. Okay. Here on 2nd carbon and 4th carbon, oxygens are present. 2 oxygens are present. That's the reason why 2,4-dioxy. And on 5th carbon, one methyl group is present. 1, 2, 3, 4, 5. On 5th carbon, one methyl group is present, so that the chemical name of thymine is 2,4-dioxy 5-methylpyrimidine. Thymine is also known as 2,4-dioxy 5-methylpyrimidine. Coming to the cytosine, it is also known as 2-oxy 4-aminopyrimidine. Here you can see this is the first position. On 1st and 3rd positions, nitrogens are present in place of carbon. 1st, 2nd, 3rd, 4th, 5th, 6th. Okay. On 2nd carbon, oxygen is present, so that's the reason why 2-oxy. And on 4th carbon, one amino group is present, that's the reason why 4-aminopyrimidine. Cytosine is also known as 2-oxy 4-aminopyrimidine. Coming to the uracil, uracil is also known as 2,4-dioxypyrimidine. On 2nd carbon and 4th carbon, oxygen molecules are present, hence it is known as 2,4-dioxypyrimidine. So these are the 3 types of pyrimides that are present in nucleic acid. Thymine always pairs with adenine, as I said earlier. Thymine always pairs with adenine with 2 hydrogen bonds. Cytosine always pairs with guanine with 3 hydrogen bonds. And this uracil is present in RNA. This is not found in DNA. Okay, these are the 3 different types of pyrimidines. So okay, and now we are going to discuss about the nucleosides. Let us discuss about the nucleosides. The combination of nitrogen base and pentose sugar constitute the nucleoside. The combination of nitrogen base and pentose sugar constitute the nucleosides. In purines, the nitrogen in the 9th position binds with the first carbon of the pentose sugar. Therefore, this bond is called N9C1 glycosidic bond. In purines, the nitrogen at the 9th position forms a glycosidic bond with first carbon of pentose sugar. Therefore, this bond is called N9C1 glycosidic bond. In pyrimidines, the nitrogen at the first position form glycosidic bond with first carbon of pentose sugar. This bond is called N1C1 glycosidic bond. In pyrimidines, the nitrogen at the first position forms a bond with first carbon of the pentose sugar. These are called N1C1 glycosidic bonds. Purines form N9 or binds with the pentose sugars with the N9C1 glycosidic bonds, whereas pyrimidines binds with pentose sugar with N1C1 glycosidic bonds. Here you can see in the picture very clearly, this is the pentose sugar, ribose sugar particularly, ribose sugar, and this one is aminopurine, 6-aminopurine or adenine, and this is the nitrogen which is at 9th position, which is an imidazole ring, right? And this is the first carbon, this particular place. This is the first carbon, and this is the nitrogen at 9th position. A glycosidic bond is formed in between the nitrogen and carbon. The nitrogen is at 9th position of nitrogen base, and the carbon is at the first position of pentose sugar. And this is the glycosidic bond. This one is the glycosidic bond. So that this bond is called N9 C1 glycosidic bond. N9C1 glycosidic bond. Purines. In purines, the nitrogen at the 9th position finds glycosidic bond with first carbon of pentose sugar. Okay, so that it is called N9C1 glycosidic bond. Coming to the pyrimidines, they form N1C1 glycosidic bonds. Here you can see in the picture very clearly. And this is the nitrogen which is present in the first position of pyrimidine ring of this pyrimidine, and here you can see this is the first carbon of pentose sugar. So in this way, a glycosidic bond is formed in between the nitrogen which is at first position and the carbon which is at the first position of pentose sugar. In this way, this bond is formed. Hence, this bond is called N1C1 glycosidic bond. N1C1 glycosidic bond. Combination of nitrogen base and pentose sugar form the nucleosides. Purines binds with pentose sugars with N9C1 glycosidic bonds, whereas pyrimidines binds with pentose sugars with N1C1 glycosidic bonds. In this way, nucleosides are formed. Next, we are going to discuss about the types of nucleosides which are found in DNA and RNA. Okay. Let us discuss about the types of nucleosides found in RNA and DNA. First, we are going to discuss about the new types of nucleosides found in RNA or ribonucleic acid. Ribonucleic acid. In ribonucleic acid or RNA, the sugar is ribose sugar, and four different types of nitrogen bases are present. So in this way, four nitrogen bases form four different types of nucleosides. Nitrogen base binds with ribose sugar and form ribonucleosides in RNA. Nitrogen bases binds with ribose sugar and form ribonucleosides or ribosides. What are the four ribosides which are found in RNA? Let us see. Here, there are four different types of nitrogen bases form four different types of nucleosides in RNA. The four nitrogen bases are adenine, guanine, cytosine, uracil. Adenine, when binds with ribose sugar, forms a ribonucleoside, that is adenosine. Adenine forms adenosine. When guanine binds with ribose sugar, forms a ribonucleoside, that is guanosine. Guanosine. Cytosine, when binds with ribose sugar, forms a ribonucleoside, that is cytidine. Uracil binds with ribose sugar and form uridine. In this way, four different types of ribonucleosides are found in RNA: adenosine, guanosine, cytidine, and uridine. Let us discuss about the nucleosides found in DNA. In DNA, the sugar is deoxyribose sugar. When deoxyribose sugar binds with nitrogen base, it forms a nucleoside, that is deoxyriboside. In RNA, that is ribonucleoside, whereas in DNA it is deoxyribonucleoside, as the sugar is deoxyribose sugar. Here also four different types of deoxyribonucleosides are found in DNA. They are deoxyadenosine, deoxyguanosine, deoxycytidine, you can say, and deoxythymidine. Deoxycytidine and deoxythymidine. Adenine, when binds with deoxyribose sugar, it forms deoxyadenosine. Guanine, when binds with deoxyribose sugar, it forms deoxyguanosine. Cytosine, when binds with deoxyribose sugar, it forms deoxycytidine. Here printing mistake, sorry for the printing mistake. Deoxycytidine. When thymine binds with the deoxyribose sugar, it forms a nucleoside, deoxyriboside, that is deoxythymidine. In this way, four different nucleosides are present in DNA. Four different types of nucleosides are found in both RNA and DNA. In RNA, the ribonucleosides are adenosine, guanosine, cytidine, uridine. In DNA, the four deoxyribonucleosides are deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. Now let us discuss about the nucleotides. Let us discuss about the nucleotides. Nucleotides are the fundamental units of a polynucleotide chain. Nucleotides are phosphoric esters of nucleosides. Nucleotides are phosphoric esters of nucleosides. When a phosphate is attached to the fifth carbon, of pentose sugar of a nucleoside, a nucleotide is formed when a phosphate is attached to the fifth carbon of pentose sugar of a nucleoside. A nucleotide is formed. Usually, phosphate form ester bond with pentose sugar. With the fifth carbon of the pentose sugar, phosphate binds with the fifth carbon of pentose sugar of nucleoside and form a nucleotide. Here you can see in the picture very clearly. This part is the nucleoside and this one is the pentose sugar of the nucleoside, and this is the fifth carbon. This one is the fifth carbon of this nucleoside, and this one is the phosphate. This one is the phosphate. A ester bond is formed in between the oxygen and fifth carbon of this sugar. In this way, a nucleotide is formed. This is a nucleotide. When a phosphate is attached to the fifth carbon of the pentose sugar of a nucleoside, this is the nucleoside, this is the pentose sugar, and this is the fifth carbon. This one is the fifth carbon. This one is the phosphate group. Ester bond is formed in between phosphate group and fifth carbon of this pentose sugar. In this way, a nucleotide is formed. A nucleotide is formed. Now, next we are going to discuss about the types of nucleotides found in DNA and RNA. Types of nucleotides found in both DNA and RNA. First, we are going to discuss about the types of nucleotides found in RNA. When the nucleoside of RNA binds with phosphate group, a nucleotide is formed that is called ribonucleotide. A ribonucleoside binds with phosphate group with ester bond, forms a nucleotide that is called ribonucleotide. 4 different types of ribonucleotides are found in RNA. They are: when adenosine is a ribonucleoside, binds with phosphate group, forms a nucleotide that is adenylic acid. When phosphate is attached to the nucleoside, it impart acidic property. Hence here we add acid. Adenosine is the ribonucleoside. When it binds with phosphate, it forms a ribonucleotide that is adenylic acid. Guanosine is another ribonucleoside of RNA. When it binds with phosphate group, it forms a ribonucleotide that is guanylic acid. Cytidine, here spelling mistake. Cytidine, when binds with phosphate, it forms a ribonucleotide that is cytidyllic acid. And the uridine is the another ribonucleotide of RNA. When it binds with phosphate group, it forms a ribonucleotide that is uridylic acid. So these are the 4 different types of nucleotides that are found repeatedly in the RNA. They are adenylic acid, guanylic acid, cytidyllic acid, uridylic acid. Now let us see the nucleotides that are found in DNA. The nucleosides of DNA are called deoxyribonucleosides. When these deoxyribonucleosides binds with phosphate group, they form a nucleotide that is called deoxyribotide or deoxyribonucleotide. Let us see what are the different types of nucleotides found in DNA. So these are the 4 different types of nucleotides found in DNA. Deoxyadenosine is a deoxyribonucleoside. When it binds with phosphate group, it forms a nucleotide, that is deoxyadenilic acid. Deoxyadenilic acid. And second deoxyriboside is deoxyguanosine. Deoxyguanosine, when it binds with phosphate group, it forms a deoxyribotide, that is deoxyguanilic acid. Deoxyguanilic acid. And another deoxyribonucleoside is deoxycytosine. Deoxycytosine or deoxycytidine. When it binds with phosphate group, it forms a deoxyribotide, that is deoxycytidilic acid. Deoxycytidilic acid. And fourth deoxyriboside is deoxythymidine. Deoxythymidine. When it binds with phosphate group, it forms a deoxyribonucleotide, that is deoxythymidilic acid. Deoxythymidilic acid. In this way, four types of deoxyribonucleotides are found in DNA. They are deoxyadenilic acid, deoxyguanilic acid, deoxycytidilic acid, deoxythymidilic acid. And these four deoxyribonucleotides are repeatedly found in the polynucleotide chain of DNA. These are the fundamental units of polynucleotide chain of DNA. In this way, four different types of nucleotides are found in both RNA and DNA. These ribonucleotides are covalently linked to one another to form a linear polymer strand. These nucleotides, both in RNA and DNA, these nucleotides linked to one another and form a linear polymer or strand. This is known as polynucleotide strand. Polynucleotide strand. A strand which is made up of many nucleotides. Many nucleotides, hence it is called polynucleotide strand. In next, we are going to discuss about how polynucleotide strand is formed. Okay. Now let us discuss about the formation of polynucleotide strand, a strand which is made up of many nucleotides. In case of DNA, deoxyribonucleotides are covalently linked to one another to form a linear polymer or strand, which is known as polynucleotide strand. Here you can see in the picture. And this is the nucleotide deoxyadenilic acid. Deoxyadenilic acid. And this is another nucleotide. This is deoxyguanilic acid. And this one is the another nucleotide. This one is the another nucleotide, that is deoxythymidilic acid. Deoxythymidilic acid. And this one is the deoxycytidilic acid. And these nucleotides are covalently linked each other or to one another and form a chain-like structure or linear chain-like structure. This is what we call polynucleotide chain or strand. Polynucleotide chain or strand. Like I said earlier, in phosphate group, 3 OH groups are found, in which 2 OH groups are involved in the formation of phosphodiester bond. So here you can see this is the ester bond, this is another ester bond. In this way, a phosphodiester bond is formed in between the sugar molecule and phosphate molecule. The phosphate group is linked with the fifth carbon of one sugar. Fifth carbon, this is the fifth carbon of this sugar, and the third carbon of this sugar. Phosphate moiety binds with the fifth carbon of one sugar molecule and the third carbon of other sugar molecule. In this way, a phosphodiester bond is formed, which forms the backbone. So this is the backbone. Phosphodiester bond forms the backbone of nucleotide chain. In this way, nucleotides are bound to one another and form a linear strand or linear chain or linear polymer. A polymer is formed. Polynucleotide polymer is formed. So in this way, a polynucleotide chain is formed. Two polynucleotide chains arranged parallel to each other, and complementary base pairing takes place in between the nitrogen bases of two opposite polynucleotide strands. Adenine pairs with the thymine with two hydrogen bonds, or vice versa. Guanosine pairs with three hydrogen bonds, or vice versa. In this way, two polynucleotides are bind to each other with hydrogen bonds. In this way, a DNA is constructed. In next video, we are going to discuss about the double helix structure of DNA based on these fundamentals, which we are discussing in this video, are very much helpful to understand the structure of DNA. In my next video, I am going to discuss about the structure of DNA, which is proposed by Watson and Crick. Okay. Right now, I conclude my presentation here. Thank you. Thank you one and all for patiently listening or watching my presentation and video. We will meet in my next video. Thank you. Thank you one and all.